guys, welcome to Bio 112 Online, your home for biology education. Today's topic, cardiac function. And so to talk about that, we first need to think about the heart. The heart is an organ comprised mostly of cardiac muscles, about the size of your fist, right here in your chest behind the sternum. It has two main zones. At the top, you have two chambers referred to as the atria. These two chambers have thin walls and receive blood from the veins. As they contract, they force blood into the ventricles. The ventricles are going to be the bottom part of the heart. Again, two chambers that have thicker walls, and when they contract, they pump blood into the arteries. We can divide this heart into two sides. On the right, you have the right atrium, which will take blood from the body. It will then pump it into the right ventricle. When it contracts, it will pump blood to the lungs. Returning from the lungs will be blood that enters the left atrium, is pumped into the left ventricle, which will then contract and pump that blood to the rest of the body. Now, as you're looking at this, you'll say to yourself, Dr. O'Brien, the right and left is on the wrong side. That's in part because whenever we're talking about the heart in terms of right and left, we're talking about the patient's or the heart's right and left side. You as the observer will have to flip them around in your head as you go. Between these chambers of the heart, the left and right ventricles and left and right atria, we have a series of valves. These cardiac valves are structures that prevent the backflow of blood during contraction. When they are opened from one side, they allow blood to flow, but however, if the blood is flowing in the opposite direction, then they will close and prevent it from moving backwards. The first of these that we want to talk about are the atrioventricular valves, or the AV valves. These are found, as the name suggests, between each of the atrium and the ventricle. These will open during most of the time that the heart is at rest. However, whenever the ventricles contract, those AV valves will close by the pressure of that ventricular contraction. This stops blood from flowing from the ventricles back into the atria. The second set of valves that we want to talk about are going to be the semilunar valves. The semilunar valves are located at the exits of the ventricles as they move into the arteries. The force of the ventricular contraction will open these and will force blood from the ventricles into the arteries. However, when the ventricles are at rest, the pressure in the arteries is actually higher than that of the ventricles, and the semilunar valves will close, preventing backflow of blood from the arteries back into the heart. These will all come into action as we start talking about the cardiac cycle. The cardiac cycle is going to be the rhythmic cycle of heart contractions and relaxations. In order to understand this, we have two main terms that we have to talk about. The first of these is systole. Systole is going to be the stage where the chamber of the heart contracts and is in the act of pumping blood. The second term we have to describe is diastole. Diastole is going to be the stage of the chamber of the heart when it is relaxed and slowly filling with blood. So if we think about the heart and take a look at those inner chambers, an initial point of rest will have atrial and ventricular diastole. Both the atria and the ventricles are at rest. In this phase, blood returning from the veins flows freely into the atria and the ventricles. At this time, the AV valves are open, but the semilunar valves are closed. The next stage is referred to as atrial systole, ventricular diastole. In this, as the name would suggest, the atria briefly contract, pumping the blood inside those atria into the ventricles. This force of additional blood into the ventricles causes those ventricles to supersize and grow slightly larger than they normally would at rest. The next stage would then be ventricular systole with atrial diastole. During ventricular systole, the ventricles contract. Now recall that these ventricular chambers have much thicker walls and much stronger muscles surrounding them. As a result, they go through and pump the blood with a large amount of force. The AV valves are forced closed by this intense pressure, but the semilunar valves are forced to open. The blood is then pumped into the arteries. During this time, the atria themselves are going to be in diastole and are beginning to fill with blood again from the veins. The cardiac output is going to be the volume of blood that each ventricle pumps per minute, and is going to be the product of the heart rate, or the number of beats per minute, multiplied by the stroke volume, or the total amount of blood that's pumped with each contraction of the ventricles. Put together, the rate per minute of contractions times the number or the volume of blood per contraction gives you the volume of blood per unit time, or the cardiac output. So as we go through and are thinking about this, the normal description we might have for the beating of a heart, if you were to listen to it, is this pattern of lub-dub, lub-dub, lub-dub. As we might hear listening to our own heart or listening to the heart of someone else. What we are hearing is not the beating of the heart, but the closing of the valves of the heart. So that as the ventricles contract, you have the slamming shut of the AV valves. Once the ventricles relax and enter diastole, then the semilunar valves now close. And that's the pattern that we hear. Lub-dub, lub-dub, AV valve, semilunar valve, AV valve, semilunar valve. So what we want to do is think about this in terms of the beating of the heart. First diastole, then atrial systole, then ventricular systole, then diastole once again, atrial systole, ventricular systole, then diastole once again, blood flowing into the heart, being squeezed into the ventricles, being squeezed into the arteries, flowing again into the heart, squeezed into the ventricles, squeezed into the arteries, leading to blood circulating throughout your body. To understand how this process is coordinated within this structure of muscle, we have to take a look next at the cellular level and understand how heart contractions occur at that level of a single cardiac cell. Now remember, cardiac muscle is going to be short striated muscle cells. They're going to be different from normal skeletal muscle in that first, they are branched, unlike normal skeletal muscle, and second, they have these specialized zones between each cell. They're referred to as intercalated discs. These intercalated discs are going to be composed of desmosomes and gap junctions. Desmosomes, as you will remember, are going to be basically rivets holding cells together, while gap junctions are going to be proteins that allow the free movement of cytosol and ions and solutes between adjacent cells. Those intercalated discs are going to be critical for this because they're going to allow for the information to pass from cell to cell to cell and allow these muscle cells to synchronize. Also, as a reminder about muscle fibers, muscle fiber contractions are due to the interaction of actin and myosin proteins. During muscle contractions, myosin cross bridges will bind to the actin microfilaments. In the process, they'll spend ATP, and this pulling of the myosin cross bridges is really what causes the muscle fibers to contract. However, under normal circumstances, tropomyosin will block the binding site on the actin so that the actin and myosin cannot interact because the cross bridges cannot bind on to those actin binding sites. This will then prevent the muscle from contracting. However, this will change when calcium is present. When calcium is present, then the calcium will cause the tropomyosin to shift out of the way, allowing myosin cross bridges to then bind to the actin. So the take-home message from this is that the increased amounts of calcium in the cytosol will cause the muscle cells to contract. Calcium ions are maintained at low concentrations in the cytosol of the cell. This is because calcium is actively pumped out of the cell using calcium pumps and sodium-calcium exchangers. Calcium is also being pumped into the endoplasmic reticulum. As a reminder, the ER in muscle cells is referred to as the sarcoplasmic reticulum because it's structurally different from the ER that we might find in, say, an epithelial cell. We'll use the term sarcoplasmic reticulum for the rest of this video. So here we are. We have our heart cell ready to contract, and all we need to have that contraction take place is calcium. But we just pumped all of our calcium out of the cytosol and into the sarcoplasmic reticulum, or outside of the cell. So what triggers the contraction? The cell membrane, and we'll find some old friends. The sarcolemma, or the cell membrane of the cardiac muscle cell, contains sodium-potassium pumps. Well, a lot of cells contain sodium-potassium pumps. They have sodium and potassium leak channels. They have sodium and potassium voltage-gated channels. I think you see where we're going with this, especially when we include the next one of the calcium voltage-gated channels. So these cardiac muscle cells are set up very similar to neurons. They have a sodium-potassium pump that will pump sodiums out of the cell and then pump potassiums into the cell, setting up an ion gradient. There will be potassium leak channels, which allow potassium out, setting up a resting potential, and then a little bit of sodium leaking back through to keep that resting potential off the Nernst equation. If we have an influx of sodium into a region near that cell membrane, it can cancel out some of that charge. It results in a depolarization. Because that depolarization, you can then have the opening of sodium voltage-gated channels. At some point thereafter, you can have the opening of potassium voltage-gated channels, restoring the polarization. Add onto that the depolarization will also lead to the opening of calcium voltage-gated channels. As those calcium voltage-gated channels open, they will allow calcium into the cell. The influx of calcium ions from the cell membrane also stimulates the release of additional calcium ions through the sarcoplasmic reticulum through a process called calcium-induced calcium release. So therefore, as we have a depolarization, an opening of those calcium voltage-gated channels on the cell membrane, you subsequently then have an opening of calcium channels on the sarcoplasmic reticulum. That increase in calcium therefore leads to contraction of the muscle cell. So where does this depolarization come from? It comes from the addition of sodiums from the next cell that comes in via the intercalated discs and the gap junctions on them. So if we have a couple cells, we can see the influx of sodium leading to a depolarization. That depolarization will spread from one cell to the next through those intercalated discs. We then have the opening of the calcium voltage-gated channels, allowing for contraction of those cells. But keep in mind that that contraction is happening at a different time frame than the depolarization. Recall that an action potential at one location may only last a few milliseconds, whereas the influx of calcium may last 200 milliseconds. As a result, you have this race of the depolarization and the action potential zipping along from cell to cell to cell, resulting in all of the cells taking in calcium at effectively the same time, leading to contraction of not just a cell, but the entire muscle fiber almost in unison. This depolarization then moves as a continuous wave from cell to cell to cell, providing the signal necessary to coordinate muscle contractions in the heart and allowing the heart to pump blood throughout the body. But that is the topic of the next video. So until then, stay safe, stay healthy, and I'll see you later. Expecting Thanos.